Right now, we are experiencing a historical landmark that will be reflected on years and years to come. That's right, we are living through the COVID-19 pandemic, which has taken the world by storm, or should I say infection. Severe acute respiratory-related coronavirus 2, now that's a mouthful, is a strain of virus responsible for causing COVID-19. But I'm here to explain the simple yet extremely effective genetic makeup of the coronavirus and how it so efficiently manages to hijack our cells. First, we must understand that the coronavirus's viral genome encodes for only 4 structural proteins. Let's start with the E protein, also known as the envelope protein. It starts to kick into action once the host cell is infected and is responsible for the assembly of virions, which are complete virus particles. The M protein, or membrane protein, makes up the shape of the virus's viral envelope and is the head of virus assembly as it interacts with all the other structural proteins. It's like the master operator in this virus. When it comes to the actual infection, together the M and E protein basically turn the host cell into little workshops for virus replication. The N protein, or nucleocapsid protein, is responsible for binding to the RNA genome, that when released within a host cell after being infected, acts as the set of instructions telling it what to do. It also makes up the nucleocapsid and is responsible for the assembly of virions, like the E protein. These 3 structural proteins work hand in hand together. The membrane and envelope protein make up the viral envelope and aid in virus replication, while the membrane and nucleocapsid protein work together to stabilize the nucleocapsid and aid in virus assembly. This next protein works somewhat solo to achieve one of the most important goals of a virus, latching onto a host cell. The S protein, or spike protein, is what gives coronavirus its name, as the spiky projections resemble a crown, and corona in Latin means, yep, you guessed it, crown. The S protein uses its receptor binding domains, RBDs, to latch onto and recognize a special receptor on our human cells, the ACE2 receptor. They need to find these special receptors because a virus can't just latch on anywhere, it needs to find the right place to attach so it can successfully hijack the cell. To put this into perspective, the virus's spike proteins act as keys looking for the correct lock to go into. The ACE2 receptor found in our lung and gut cells happens to have that correct lock. So now I've explained the virus's 4 proteins: the envelope protein, the membrane protein, the nucleocapsid protein, and the spike protein. Although the definitions of what these proteins do may be lengthy, it's important to realize that there are only 4 parts to this virus. The virus is crafted so simply yet effectively that it only needs 4 parts to function properly. To put this into perspective, this pen needs about 5 parts to function properly, yet it can't even come close to the damage the coronavirus can. This virus, regardless of how deadly, is so perfectly made that our own bodies, consisting of however many parts, still has a difficult time fighting it off. But by continuing to research these 4 structural proteins, we can learn so much more about how the virus wreaks havoc with its own perfectly crafted framework.